Sure.
more information on that?
Sure. So the first thing that we need to choose expedition sites well is that we need good mapping information about the seafloor. So we need to know what the seafloor looks like. So we use what's called multi-beam mapping, which is using sound to create an image of the seafloor. And it's called, we look at the bathymetry, which is like looking at like a topographic map on land, but it's the bottom of the ocean. And we can also get information about how hard the seafloor is, and we look at that with also the slope, so how kind of mountainous the seafloor is, or how steep the slope is, and we're generally looking for, sometimes we are interested in surveying the soft mud habitat, and sometimes we're interested in looking at the hard substrate. The hard substrate is where we find coral and sponge communities, which can often be something we're interested in looking at. And then we can pick those dive sites based on that, also based on any previous cruises that we've done or previous surveys. Sorry, I'm getting distracted by this rock, and there's a nudibranch right there in the middle.
Oh, I love nudibranchs.
And then there's also management issues that we're trying to address. And
are those the potential sponges?
So that was what I was interested in looking at, and I'm not sure if that's it. And then I also see that they made a collection already on this dive of some of these, so I don't.
Okay.
I think they already collected these.
All right, coming wide.
So for example, in this dive, we're looking at areas where their fishing management changes have been proposed by the Pacific Fisheries Management Council. They review periodically what regulations are in place and whether they should open or close areas to fishing. And some areas that we're looking at here have had proposed changes to be opened or to remain closed. So we are collecting imagery in those areas so that we can assess what the communities are like there, because they haven't actually been visually surveyed before. And then once changes are made, we can see how those might change, or if we can provide information to inform those decisions that are made.
Mm-hmm. Yeah, that applied piece, I think, is just really, important for people to understand how we're using this science to really make management decisions as well.
And the EV Nautilus, the exploration vessel Nautilus, really is well equipped to explore. So we often select places in Cordell Bank and Greater Farallones National Marine Sanctuary. We're focusing on places where we haven't been before so that we can explore and look at some of these areas for the very first time.
Very cool. Taylor, and because there's just so many high school groups on right now, I think it's really cool if you could share a little bit about your story and this internship program and how you got involved with EV Nautilus.
Yeah, sure. So I actually grew up in the Midwest. I went to high school in the south suburbs of Chicago. I always was interested in science in high school. I think I was about average, never really took much interest until I think college, when I really had to figure out what I wanted to do. So I went to Ball State University in Muncie, Indiana, and there I got an undergraduate degree in general biology with the goal of studying marine science at some point. It took me a few years to finally bridge the gap between the Midwest and the ocean life, but after a few years of volunteering and working in an algal ecology lab, doing really well in my classes, participating in aquatic fisheries stuff, I was able to earn a scholarship to attend Duke Marine Lab, where I was able to spend a semester there and do my own independent research and take some courses and really make a community for myself with people that were really interested in what I wanted to do. I hadn't really seen many people like me, a woman of color in science, doing marine science. So it was really uplifting to meet all these different kinds of people. So in high school, you might not know what you want to do yet. But just explore these options that you have. Google things, ask people questions. Don't be afraid to ask questions. I ask lots of questions. I don't know everything. And you can learn a lot. Even if you are told you're wrong about certain things, that's okay. It just helps you navigate to where the right place is for you. So high school is a time for you to learn a lot about yourself and enjoy it. It's okay to be different. I'm definitely somebody who's very unique, but now I'm seeing that that's something that's valuable and not something that you should feel ashamed about. So yeah, here I am as an ocean sciences intern aboard one of the most amazing vessels with Ocean Exploration Trust and learning a lot here. So I'm really, really thankful and blessed to be here. So yeah, just keep working hard and figuring out what you like to do, and if this is something you're interested in, there's definitely opportunities to get involved. You can educate yourself using online tools, asking questions, reaching out, even to people like me, finding their emails and just asking them questions and what they kind of did to find their way to where they are. I'm sure most people are happy to help and give you some advice.
Thank you so much for sharing.
Of course.
Yeah, I think something that was really encouraging for me to hear prior to even getting on the boat was I had another past Nautilus intern come to a marine science festival that I was hosting in New York City, and she was just saying that once she interned on the Nautilus, just how many doors that opened, and the confidence that she gained from that experience, and then all the connections she made through kind of just putting herself out there, sending that email to that scientist or that researcher and saying, hey, I had this, this experience, I'd love to get more. And she just said that she's really been encouraged by the, building blocks in her career with the EV Nautilus experience under her belt. So.
Yeah, it's a great place to meet amazing people from all different types of backgrounds and pathways and roadways that they use to get here. So it's a really, really diverse, I guess, group of people that come here and share their skills with everybody.
Totally, yeah. So to make this work happen, I mean, it's 31 scientists. We're divided into 3 watch teams of about, 8 people or so. And we have people that are engineers and ROV pilots and expert mappers in GIS video engineers, videographers, scientists of many different types, biologists, geologists, a coral specialist here from Cal Academy of Sciences. So this team of experts, we're collaborating to bring you guys, down to the ocean floor and to learn about these amazing national marine sanctuaries on this expedition. And then also on the vessel, we have a team of, a crew, so a captain and mates that are helping us navigate and stay safe on the vessel. And
are we waiting on the ship?
And we also have a team of culinary staff cooking us amazing meals that we all need to power through our watch schedule. So it's very much a team effort and a very diverse team. Oh, excuse me, and also data professionals, and a very important piece of this puzzle.
It's a great way to captivate the diversity of people involved with carrying out a mission. And it's really those diversity of skills that make it successful. Not everybody can have every single skill, but working together, a lot more things can happen.
Miles is close, though.
Sorry?
I said Miles is close to having every single skill.
Oh, yeah.
Oh, thank you.
Miles, remind us what number cruise is this for you?
Aboard Nautilus, it's around 35. Total, it's around 44, I think. I had it counted up earlier this year.
So fun.
That's so neat.
We've been traveling along this transect line. We're getting close to an area that looks kind of interesting after A1AAE to 1ABS. Looks like it could have some higher slope habitat, so that is our destination at the moment.
Nice close-up of a crinoid, a feather star.
Behind it is this encrusting tunicate that we've been seeing in Greater Farallones National Marine Sanctuary and Cordell Bank National Marine Sanctuary, and we actually collected some last year. Actually, could we zoom in? We can see some of the structures inside.
I love these zooms because we really get to see more of the other relationships of species on the rock. Seeing some krill. I see a tiny little crab underneath that crinoid arm.
Okay. Each one of these features looks like it could be, you could spend an hour on it.
Damien from Romania was curious about marine debris. There's a lot of talk in media about the amount of plastic in our waterways, and Damien would mention that we're not seeing too much on the ocean floor. So we've come across a few large pieces of marine debris. We had a 5-gallon bucket, a tire, a very clear soft plastic Ziploc bag. So we've seen some. And of course we know from research that microplastics are not only in our surface areas, but also on our seafloor and in our benthic zone. So we're seeing impacts, but
Some cup
you're
corals.
right. Thankfully not too many examples.
On the, based on the science loggers observations, we're averaging 2+ trash observations for the entire West Coast dives for all of Nautilus.
Oh, sorry, 2+ per dive.
Per dive.
Thank you.
So one of these cup corals has its polyps pull in, but the one behind the fish has it out, I think.
Under his chin.
Yeah.
It's his pillow. It's neat to see the light going through the clear part of his eye. You see how it's focusing the light onto his cheek? That's so cool.
That's a sharp chin. Great, thanks.
Okay.
Can really see the polyps on the other one below it too. A lot of people don't realize that we do have corals here. As an educator at the sanctuary, so many people are just absolutely surprised to hear about corals in this cold, deep environment that is just so full of plankton, but that's the deep water corals. That's what they thrive in. We may not have tropical systems here, but there are other parts of the planet with that. Here's a nice lingcod. And deep water corals rely on food that rains down from above, and they use their polyps.
Octopus.
Uh-oh. I think we're gonna. There he is.
Ratfish.
That octopus is like,
On the
you don't
move.
see me.
I'm out of here.
He's on the move.
Wait, is this my?
No.
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 The coloring.
No, sorry, Gary.
No,
Shucks.
no.
It's a small one though.
Yeah, it's still cute. It's World Octopus
You're
Day.
about to hit the end of
Yeah,
Gosh,
Taylor,
it
for
is.
remembering.
Happy International Octopus Day. Thank you, Taylor.
Today's your
Ann.
day.
How's it going?
He's like, I was chilling. And then
Yeah.
he reminded me it's time to party.
I think this might be a red octopus.
That's what I was thinking too.
Yeah,
I was just gonna look that up to confirm.
It's like, I'm not committing to putting all my tentacles out. Just too risky.
Okay, hold on. We can definitely get some more contrast. There we go. There we go.
I feel like we're getting back to a lot of krill.
Yeah.
Oh yeah.
It's got an orange eye. Okay. I think,
Wayne, could you put the focus on the krill?
sure. Focus near. Let's see.
These krill are just a huge staple for the food web here, not just for whales and some seabirds, but many rockfish. And as we observed yesterday, sea stars eat krill as well. These big sea stars on the floor, Rathbanaster with lots of arms. We saw them, the arms up catching krill, which was really neat to see.
Okay. Looks like the krill have gone back to the seafloor and wide.
Is that a cowcod?
I don't think so.
Stripes.
And spots.
There's a skate to the right. The flatfish. Can you spin to the left a bit? There's kind of a large feature. Okay, thank you.
Someone tuning in was curious about opportunities for some of our older community or senior citizen community in participating in ocean exploration.
I think by watching at home and sharing with friends and family, educating others is a part of ocean exploration that we need more of. More people to be talking about these unique environments and the value of them and supporting research and science. This is one of the nice things about Nautilus Live is that we can bring it to everybody. And being able to follow along at home. I've always wondered if there's Nautilus Live viewing parties. I've thought about that, but I can't quite get my friends involved. They just haven't been into it as much. But such a great opportunity for everybody to participate at home and to share the uniqueness and of these species and the observations with other people. Writing about it with larger audiences. It's never too late to go back to school. My friend Carol Kuiper went back to graduate school in her 50s and spent a good portion of her older years in science and marine science because she just loved it so much.
Yeah, there's definitely no age limit on any of the opportunities that are available on EV Nautilus or with the sanctuary. So we are happy to have everyone of any age help support us.
For sure. I know in New York City, there's tons of programs around testing water quality, especially at locations where people access the water for recreational reasons. There's a lot of oyster restoration, and sometimes that just means helping to clean off cages or help count live oysters. But the more hands can help to move a project along more efficiently and therefore help us
Let's
to...
look at the side of that rock with all those. That might be zoanthids. I thought maybe it was cup corals at first, but I think it's zoanthids.
Yeah, copy
So, yeah.
coming around.
Look up citizen science in your area or community science to see if there's projects.
And for those that are local, we are here in Greater Farallones National Marine Sanctuary. Oh, here's an egg bundle on the side
Cool.
here. So
Oh,
this is,
yeah.
we've seen these before that ended up being cat shark eggs.
Okay.
I'm not sure if that's what these are, but they end up being all bundled together like that.
Very neat.
Greater Farallones National Marine Sanctuary has a great citizen science program that many seniors, retired folks, and people who have moved on to the next portion of their next chapter after their careers have gotten involved in. It's called Beach Watch, and they survey local beaches once a month and record live and dead animals and characteristics about the beach, and it really helps understand those ecosystems and becomes important when there's things like oil spills or other events, and they can see how those environments looked before and after. They also find events like when there are beached marine mammals or strandings or die-offs of seabirds, and they're really the first feet on the ground being able to pick up some of those signals early on.
That program's also been helpful for folks who wanted to transition in careers and getting experience
Well, there is a
marine
cup
science.
coral in among those, but kind of a mix. Yeah.
Oh, there it is. I see it. That orangey one's more orange.
That guy.
Yep.
Yeah.
That's it.
Okay. Lots going on here.
Yeah.
We have a very specific question from someone named Connor. They're tuning in from a university of, I
Great.
believe, Leiden. The question is about sponge samples. There's curiosity around if sponge samples are used for DNA barcoding of the microbiome in sponges.
Oh, that's a good question. There might be that kind of work going on somewhere that I don't know about. What we are trying to do is we are collecting sponges to, we really don't know a lot about the species of sponges. They haven't been well described. So a lot of these species that we look at, we describe by morphology. This is a vase sponge, this is a barrel sponge, but we don't actually know what the species is. So we are collecting sponge samples to look at try and figure out what species they are. And then we are also looking at the DNA of those sponges so that when we collect eDNA samples, we know if there's DNA from sponges in there. We don't know what their DNA looks like all that well. So we are looking at the DNA of the sponges, but I don't know about work that's going on to look at other associated species within sponges, if that was what the question was.
Thank you.
So those are all the same species of anemone, they just have that color variation.
Okay.
Metridiums.
Skate egg.
Skate egg, cup coral.
Someone tuning in also offered that a lot of universities have continued education programs and even free learning opportunities for people in their community. Some are specific to senior citizens or retired folks, so something else to check out. And again, if you're listening, you're tuning into the 4 to 8 watch shift. So the Nautilus is organized into different watch teams and a watch schedule. So we're 4 to 8, meaning we are in the control van from 4 a.m. to 8 a.m. Pacific time and 4 p.m. to 8 p.m. Pacific time. And this allows for us to conduct long-term dives. So the dive we're on right now is estimated or expected to be about 22 hours in length. So our ROVs went in the water yesterday afternoon at about 4 p.m. and have been continuously at around 200 meters of depth. And since then, different watch teams have cycled through our control center, our control van, to bring you this live coverage and analysis of what we're seeing. And I think my mom might be tuning in right now. So hi, Mom. Thanks for listening.
cushion star, I think.
I was thinking also a little bit more about the citizen science and ways to be involved. This is not deep ocean exploration as an example, but in California, the California Academy of Sciences hosts these BioBlitz days during super low tides to catalog as many species as possible using a community of people coming together and having some experts help facilitate, but also using the iNaturalist application. And the California Academy of Sciences works with the data they gather from these BioBlitzes to build on species lists, build on distribution, and it's a really neat community of people that you can have people help verify your species.
There's some more egg bundles.
Yes.
Right around that sponge.
This rock that we just went over.
Ooh.
Oh yeah, right there too. So many, wow.
So is that typical for sponges to be that like densely covered in like sediment or whatever that is?
For this type of sponge, it is. So this is kind of barrel sponge. I think this is a Rhabdocalyptus. And they're always fuzzy like that. Some of them are and some never are. I'm not sure why.
But great point, Jenny, about BioBlitzes, and I know that they happen all over, so there's opportunities in many areas to participate, and we benefit from people participating. And I know in New York City, they've been especially exciting because, frankly, sometimes we forget that in New York City, where I'm from, there's a lot of nature around us. And so when we stop and collectively take a second to actually look, we can see a lot of diversity and even find new species. Ranges, at least.
And you guys can see in bubble cam, there's a rockfish in there.
Oh, yeah.
Oh my
That's
goodness,
cool.
Straight
yeah.
down.
If you look in the quad view in the bubble cam, you can see that there's a rockfish.
Maybe two.
Yeah.
Come out, come out.
That was interesting.
I don't quite have the angle to look in there what this is.
I think I would hit the porch before I saw the straight down.
There's also a bunch of seabirds sitting outside the wire view camera.
Yeah.
I'd love to get a good view on those. I might need to go take a little walk. We're surrounded by so much.
So I think I'd like to swing south a little bit to see, maybe look down slope a little and see if we hit some of this higher slope habitat that's coming up in this area.
Roger, can do.
So we're just getting to this area that looked interesting on the habitat. Yeah, this looks good. We could start running along this contour and see how it looks.
Are we scheduling
Okay.
a transact?
I just want to look around first
Okay.
and explore. We may do a transect, but I don't know if we have enough of the habitat we're looking for. So let's take a look and see what we find. So you are free to zoom, Wayne.
Oh, zooming now. You don't have to ask twice. I like that bright orange. Okay, let's see what we can actually. The textures are fantastic.
Let me back up a little and see if I
Okay.
can get the light on it.
Yeah, I can work with that. That's enough. Pretty good. I want to see this patch of textured, clear. Let me get it. Oh, super sharp. Let's see. Okay, now. Oh, it's okay.
Yeah, I don't mind.
Come
Sorry, I was trying
That's
to get
all
the
right.
tube worms in.
Yeah. Those are good. That's good. And
Can you zoom right in on one of the tube worms? Or we lose the viz?
that's what I got.
Sorry, it dusted it a bit there.
It won't last long. It'll clear out. It's like,
What is the yellow and white sphere?
We were just looking at that. It's a brachiopod, but it has something growing over the shell. So we're looking straight down on the shell. It almost looks like a ball from this angle, but it's the top of the shell.
I see.
obviously a tiny tennis ball.
Right.
See 3 skate eggs.
Oh, on the right. Yeah, I see him now. Excuse me. That's
Take
rude.
my picture. Take
That's
my picture.
rude.
Likes the limelight.
He's like, he's trying to hide something.
So cool.
Another cup coral.
Mm-hmm. Oh, in the lower left, or the left,
Yeah, left.
the little white one.
Mm-hmm. This is reminding me of an activity I wrote up years ago called Every Square Inch Counts. And it might be time to update it with some new imagery. But it's basically just an idea of how to look at seafloor habitat in the quantitative sense. We study the intertidal zone with quadrats, and in some cases we do the same with some subtidal habitats. And I put an activity together with some photos of Cordell Bank with different depths, and now I can go a lot deeper with some of our Nautilus imagery.
Yeah.
But every square inch counts, for sure. All that habitat.
Yeah.
Someone tuning in is curious about invasives, but in particular, they're referencing a tunicates, and if any encrusting tunicates are invasive or an issue in this area.
Yeah, we collected an encrusting tunicate like that on one of our last surveys last year, and so we're not sure exactly what it is yet. And there is an invasive tunicate that hasn't been identified out. It's been identified near shore, but not offshore in deep habitat off California. It's something that we're concerned about and keeping an eye out for, that we haven't seen or confirmed yet, but there is a species didymnum that we are aware of and are keeping an eye out for. As far as we know, it hasn't invaded and taken anything over yet.
I think I noticed a few parasites on that skate too.
I feel like there's a lot of small rockfish right here. Or are we just further up from the bottom?
No. I think that's correct.
Yay! A ratfish.
Yeah, the smaller fish are harder to see when they stand still. Okay. Okay. Oh, he has a friend.
Yeah, I liked watching it change direction and use its tail.
Okay. Oh, lost the focus. There we go. Oh, it looks like he's gonna settle in. Okay, coming by.
All right, so I don't know if this is exactly the habitat that we're looking for. It was the hardest, rockiest substrate we've seen, so I'm going to do a 10-minute transect here, if that works for you guys.
Okay.
Yeah, works for me.
Let's see. Okay, so let's
Me,
get
Can you remind me what these longer fish are? One was, we kind of just passed in the lower part of camera.
I did just see a
Hake?
rockfish swim off
Yeah,
screen.
it was before that.
Okay. Sorry, I
No.
wasn't looking.
What's her speed?
The sil
Liz?
Currently 0.3.
0.3.
kind of long silvery fish that are quite mobile are hake,
Hake.
if that's what you saw.
Okay,
Yeah.
yeah, possibly.
Our bearing is 132.
032.
132.
I mean 132. 032. Where'd that come from? Yeah, right here. We'll start out slow.
Your heading goal is 127.
132, you said, right?
Correct. I don't know if that changes the X and Y goal, if it's already been put in.
Yeah, I put it in. Seems to be moving. 0.2.
No, I know. When you change the heading, I don't know if the goal
Updated.
rotates with that.
Oh, and that's a cycle of. Should we speed her up a bit?
Hmm? I can. That would take a minute. Yeah, something we want to do.
That's what it is. It's another camera 9 sunrise.
Oh, that's lovely.
What is camera 9 here? I can't switch it, can I?
Yeah, you can. It's either port or aft robo.
One of these two?
Yeah. Could you put Argus on 13 then?
Why?
Why? Because I use that.
Huh?
Because I use that.
Argus?
Yeah.
It's not going to be there that long. I use that. Huh? I'll sacrifice my bubble cam for.
See, you're all stretched out.
Did we start the
that.
transact yet, or are we just getting in position?
We're waiting for the ship to speed up.
Oh, okay.
We've given her a 0.5, and I'm probably going to have to stop and wait there.
Okay. So, hi Susie in Germany. My sister's tuning in. Thanks, sis. And just to remind folks, we deployed our ROVs yesterday at 4 p.m. Pacific time, and this is anticipated to be a 22-hour dive. So likely we'll be recovering our ROVs somewhere around 2 p.m. Pacific time.
We're just going to enjoy the sunrise a minute while we're waiting for the vessel.
Yeah. Also, if anyone wants to share a highlight, or one of our viewers is asking for a memorable moment from this expedition thus far in Cordell Bank National Marine Sanctuary, the Greater Farallones National Marine Sanctuary, feel free to share.
I think I'll remember this cruise by this really amazing weather we've had out here and how calm it's been and how we were worried that we weren't going to be able to get up to this location to survey, so we've gotten a lot of work done here, so that's been memorable. And in terms of what we've seen on the seafloor, let's see, Cordell Bank National Marine Sanctuary dive a few days ago. I was really impressed by the tube worms that we saw. That was a new finding for me, a new observation in our sanctuary, so that was really neat. Been seeing a lot of octopus on the past few dives, but no flapjack octopus yet today. Today we've been seeing a lot of cat shark or skate eggs, which we've been seeing skates, and so maybe they're skate eggs. That's been a nice observation from our dive today. Lots of rockfish today, so that's been great.
How about you, Wayne?
Yeah, my favorite observation was we were zooming on a feature, a rock feature on the bottom, and there was a brightly red colored crab that was just scampering around the base, and as we got a little closer, he was frightened of us, so he kind of scampered under the rock, and as he went under the rock, an octopus arm came out over his head and gave him a hug, and that was kind of fun. So
Yeah, I love seeing those little critters and their cool behaviors.
yeah.
Taylor Ann, is there anything memorable?
I'd have to agree about the tube worms. That was my favorite. That was really cool. They were all just like huddled in little crevices of some rocky structures, and we got to see their plumes that were kind of like almost like a tulip or rose shape. They were really beautiful, as well as the giant Pacific octopus we saw. The color of that octopus was really beautiful, and we got in really close, and it was really awesome to see that. But yeah, I think almost everything is my favorite. I love seeing the diversity of life down here. We've seen so many different things, and I'm excited to see what else we find as we continue our dive. Hopefully we find some rocky slope area.
I agree. It's hard to pick one thing. Especially I'm someone who's focused on an ecosystem, so it's hard to just pick out one thing when interested in seeing the whole picture.
Okay, that's Herc, Argus, and Nautilus all on the move, approaching 0.5 knots.
Okay, we'll go ahead and start transect at 13:49:05.
Copy that. You might have to repeat one more time when the navigator gets his headset on.
Target dropped.
Ah.
Oh, yeah, thanks.
You can remember those numbers?
What number? She has time.
Sorry, Miles, I should have waited till you're in your seat.
It's all good.
So listeners, we're running a transect where we stay at a consistent speed, zoom, height above the seafloor. And how will this transect be used, Danny?
Well, we record this on the video and then take it back to the lab where we have a scientist who will be watching this video and counting all the organisms in the frame. And we've been doing a few transects like this in different substrate types. And because they're all standardized to those parameters that you mentioned, we can look at the density of animals in each of the different habitats and be able to compare them. It's a way to standardize the transects and be able to quantitatively analyze them. Previously on this dive, we've kind of been poking around, zooming in at things, just kind of zigzagging along. And so this is a way to just take a straight line and have a nice view that we can have a designated length at and quantify all the organisms in that transect. We've been on this dive for over 12 hours now, and this has been a nice stretch of some rocky habitat we're on. I see that up ahead of us, we're about to take in a few more transect lengths. We have a left turn, it looks like, out to some soft flat habitat again, so I thought we'd get one more transect in before we change habitat types.
Do you remember the name of our deepwater coral expert on the mid-Pacific Islands? What was his name?
On what cruise?
Between Hawaii and Samoa.
Oh, Steve Oskovich.
Yeah, that's right.
Yeah.
Steve has watched how many hundreds or thousands of hours of ROV video. So he was a deepwater coral expert that could spot or identify a coral from before we could even see it. But a lot of his expertise and his PhD was reviewing ROV video. I forget the number that he quoted, but it was probably more. He had watched more ROV video than we all have sitting here looking at the screen for a living. And that was part of his expertise. So all the NOAA video, Sebastian video, Nautilus video, while sitting in a dark office somewhere.
Temple.
That's amazing. So if you're watching at home and you love watching this video, you could make a career out of it.
Caitlin's on her way.
Yeah, one of our fellow scientists joining us who has another watch. It's her job, Caitlin's job, to review these transects in this video and identify and count species. So she has a wealth of knowledge.
The West Coast National Marine Sanctuaries wanted students to have the opportunity to see what that's like to do a transect and count things. So we created a curriculum called the Deep Sea Coral Curriculum that can be found on National Marine Sanctuary's website. And it's geared towards middle high school age students, but part of the activities the students can do a transect with a video clip from some of the deep sea explorations we've done, and they can compare across 5 different